Hi, I'm Chelsea Carey. At Point Blue Conservation Science, I serve as the Working Lands Research Director and Principal Soil Ecologist. In this video, I'm going to talk about an inoculant-supported restoration project that Point Blue recently undertook and provide guidance on how to conduct this kind of restoration yourself should you be interested. Healthy riparian forests provide critical services to humans and wildlife. These include clean water, protection from flooding, wildlife habitat and refugia, and climate change mitigation. Unfortunately, the majority of riparian forest habitats across California have been lost or degraded. At Point Blue, we have a long history of restoring riparian forests throughout the Bay Area as part of our Students and Teachers Restoring a Watershed program. In this program, we work with kids of all ages to conduct professional restoration grade projects that include planting native trees and shrubs to rebuild the health of these important areas. We take a climate-smart approach to our restoration projects, which means that we make sure we plant a diverse array of local species and that we include ones that have ecological traits that help them to withstand projected conditions at a site. This might include hotter and drier conditions or more frequent fires. Inoculant-supported restoration builds off our climate-smart approach by also considering the life below ground. In a handful of soil, there are more organisms than there are people on the planet, and some of these organisms form really important partnerships with plants. Mycorrhizal fungi in particular form associations with plant roots and help the plants get the water and nutrients that they need to grow. For some of the plants that are common to restorations, like oaks and willows, the mycorrhizal fungi that these plants form associations with, which are called ectomycorrhizal fungi, they may be absent or at low densities at those sites. This is especially true for sites that have been devoid of ectomycorrhizal host trees for a long time. We found that introducing whole soil inoculum that includes mycorrhizal fungi during the restoration process helped to increase germination and early survival of both coast live oak and valley oak trees. And in fact, introducing inoculum that was collected from local areas that are warmer and drier than the restoration site itself, that actually amplified the benefits seen for coast live oak and arroyo willows. Specifically, the odds of survival with heat-adapted inoculum were 4 times higher than controls for willows and 3 times higher for coast live oak. One possible reason for this is that the fungi from warmer and drier regions have traits that allow them to withstand drought or higher temperatures in the areas that they were introduced to, thus conferring greater benefits to the plant. The process of conducting inoculant-supported restoration, the way that we've done it and recommend here in this video, it includes 4 steps. In this video, I'm going to walk us generally through these 4 steps, and I encourage you to check out the companion technical report guide for more details. The first step is to identify sites from which you'd like to collect inoculum. Using local knowledge or GIS and publicly available data sets on climate and soil type, find a site that fits your criteria. In our project, we were looking for collection sites that had the target tree species of interest, had similar soil, and either similar climate or warmer and drier conditions compared to the site that was being restored. Once you've identified a location from which to collect inoculum, the next step is to make a site visit. You'll want to find 3 to 5 individual trees per host species, so in this case, we sought out coast live oak, valley oak, arroyo willow, and Doug fir. Focus in on those individuals that don't show any obvious signs of disease, and which otherwise appear to be doing all right. At each tree, you'll collect soil twice, once upslope and once downslope. You'll go ahead and take 1 pace away from the tree's trunk and use a pre-sterilized hand trowel or soil knife to collect soil from the surface down to 15 centimeters depth. You'll do this excavating a hole that is about 10 by 10 centimeters in area. Go ahead and combine the samples in a gallon-sized Ziploc bag, keeping samples from each tree separate. You can transport those back to your facilities for storage and Phytophthora testing. Now that you've collected your soil samples, you're ready to do Phytophthora testing. Phytophthora are pathogens that are responsible for killing plants throughout California. Perhaps most conspicuously, they're the culprits behind sudden oak death. Phytophthora species can spread to new sites during restoration. At Point Blue, we take every precaution to minimize the possibility of that happening, and we encourage you to do the same. A relatively simple yet effective way to test for the presence of these pathogens in plant and soil samples is to conduct a method we call pair baiting. We've adapted the testing protocol we use from an online source called Phytosphere. The basic approach here is to use green pears, Danjou pears preferably, to bait any Phytophthora that may be present in your sample and to watch for signs of disease. This entails first reviving dormant Phytophthora if necessary by moistening soil with water for a few days. Then baiting the pear by incubating it in your soil sample that has been saturated with water to the point of flooding. Be sure to mark any existing blemishes on the pear prior to addition to the soil. After about an hour, drain the leachate from the soil and continue incubating the pear in the leachate for 3 days. At the end of the 3rd day, remove the pear from the leachate and let sit for 5 more days. All the while, keep an eye out for signs of Phytophthora. You'll want to report and discard any samples that show symptoms and save the soil and leachate from those samples that were clear. These can be stored at room temperature and used as inoculant during the restoration process. In this portion of the video, I'll be joined by Jessie Ditmore, a straw project manager with Point Blue. How you install your plant is going to depend on what kind it is. For instance, planting willows typically occurs by inserting cut sprigs, whereas planting oaks is often done by burying acorns. Regardless of the plant species, the method to add whole soil inoculum is going to be similar, and this includes adding soil and leachate from pear baiting to the bottom of the planting hole. Because you're simply trying to introduce enough ectomycorrhizal fungi to colonize the plant roots, we found you don't have to introduce much inoculant. In our study, we added 2 tablespoons of soil and 1 tablespoon of leachate with each plant. You can complete your plant installations as you normally would, but now they have their added microbial partners to help them survive better in the near term and hopefully thrive in the long term. If you'd like to know more about how to conduct inoculant-supported restoration, again, I encourage you to check out our technical brief. I wish you all the best as you endeavor to perform inoculant-supported restoration as part of your next restoration project.